Thank you for the invitation to come give a talk to you. I've really enjoyed my time in Spain and meeting lots of people here. So today I'm going to talk about work on lice and looking at the history of host-parasite coevolution. And one of the, in evolutionary biology, we study processes at two fundamental levels. One is a microevolutionary scale. For example, we could study natural selection. So in different populations, does natural selection favor blue over red? And we can also study this on a this process is on a macroevolutionary scale. For example, reconstructing over a tree of species, evolutionary tree of species, when did blue arise from red? But one of the main challenges in evolutionary biology is actually making the linkage between these two scales. Can we predict macroevolutionary patterns from understanding of microevolution and ecological processes? So that's one of the fundamental questions we try to address in this work. And permanent parasites or permanent symbionts are a very good system to study this, because in this case, if you have the host, in this case the Columbolivia, the rock pigeon, you have the entire environment for the parasite. These lice spend their entire life cycle on the host. They glue their eggs to the feathers and then molt through 3 nymphal instars back to the adult. So they spend their entire life on the host, so it's a very simple system. In particular, we've been focusing on the lice of pigeons and doves in the order Columbiformes. Lice are in the order insect order Theraptera and the suborder Ichnocera, and they eat the downy parts of the feathers. So here's a body feather from a pigeon that has a very low louse load, and here's a body feather from a pigeon that has a very, very high abundance of lice, and you can see they eat away the downy parts of this feather. That interferes with the bird's ability to thermoregulate, and they have lower overwinter survival if they have a high louse load. In pigeons and doves, there are 2 forms. One we call wing lice in the genus Columbicola that hides out in the wing feathers but goes to the body to eat the downy parts of the feathers. And then body lice, Bisclenoides, which hang out on the downy on the body feathers and eat the same downy parts of the feather. So we can compare these 2 groups of parasites on the same group of hosts. And so if you look at host specificity, that is how many hosts do an individual parasite species use, we can compare these 2 groups of lice. So for body lice in the genus Bisclenoides in the New World, there are some species that are completely host specific. So Bisclenoides zeniteri only occurs on Zenita macura, the mourning dove. In Columbicola, the wing lice, the situation is a little bit different. So there's one species here, Columbicola macuri, that occurs on 7 species of doves in the New World. And so if we calculate on average how many hosts does a given species of Louse parasitize, for wing lice it's about 3 species of hosts, and for body lice about 1.8 species of hosts. But this is based on a morphological assessment of species boundaries, so we wanted, before that, so there are two potential determinants of host specificity. One is dispersal. So to be on more than one host, a species must be able to disperse between those 2 hosts, move between those 2 hosts. If they can move between the hosts, they also have to be able to establish, and this is also relevant for host switching. So they have to be able to survive and reproduce on multiple host species to be a generalist. So we can study specificity at these 2 different levels. I'm going to focus today on feather louse dispersal. So there are 2, in parasites we use the term transmission. There are 2 types of transmission. Vertical, it's a dispersal from parents to offspring at the nest. In horizontal transmission, this is dispersal between birds of a similar generation. So this can occur by direct contact between the bodies of birds. Maybe lice can move. Maybe lice fall out in dust baths, shared dust baths between different species. And there's another phenomenon called pheresis, or hitchhiking, that occurs in some lice, where if you look at hippoboscid flies, which are winged diptera, also parasites of birds, they can fly between birds. Lice are wingless, and you can find them attached to the body of these hypoboscid flies. These are sometimes called louse flies because you can catch them and find lice attached. And presumably these lice are using these flies to move between host individuals. The flies are not as host specific, so they can be on many species. And so this might be a way for lice to move between species. If we compare published observations from ecology and experiments, we can compare what is the dispersal ability of wing lice, Columbicola, and body lice, Phylogenomics. In both cases, vertical transmission is very high. So when the chicks start to develop feathers, lice will quickly move to the chicks in the nest. There are lots of records of pheresis or attachment to hypoboscid flies for Columbicola wing lice, but almost no records for body lice, and there have been experiments to show this is the case as well. And there are also several records of wing lice from doves on the Galapagos hawk, a predator of doves on the Galapagos Islands, but no cases of body lice on these hawks. So wing lice seem to have a better dispersal capability. So when we think about dispersal, we can imagine this at several different scales. So one might be just moving within an individual bird from different parts of the bird, from the body to the wings and so forth. We could also look at dispersal or population structure between two birds from the same flock and their individual populations. We could also think about dispersal between populations of birds of the same species, dispersal of parasites between populations of birds, or dispersal between different species of birds by these parasites. Okay, so the scales I'm going to focus on today are between different populations of birds and between different species of birds, looking at the population structure of these parasites. So this is a study published a long time ago now, but just using the CO1 barcoding gene from the mitochondrial DNA, looking at this widespread species Columbicola macuri that occurs on several species of doves in the New World. And what we found is very high divergences between some clusters of haplotypes. So here's one haplotype group of very similar sequences, differing by about 20% sequence divergence from these others. So very big divergence, and it only occurs on the mourning dove, Zenitis macuri, here in green. There's another group cluster here that occurs on 3 different species in the different colors. Almost no genetic structure or genetic differences there, but high genetic difference from the other haplotypes. So we found deep structure between some hosts, but then almost no structure among some others. For a different species of wing loss, Columbicola passerini, we had a similar situation where here's one cluster was only found on the blue ground dove, and here's one that was found on 2 species. Okay. We also looked at body lice, and in some cases we did have a widespread species. Here's a species on 3 hosts, and we found even more pronounced structure. So this one was only on the Inca dove, and then we even found within one of these major haplotype groups even more fine-scaled population structure between host species. And it's similar for another group of closely related Phylogenomic species, even between different locations. Here's lice collected in Mexico versus Texas. There was a genetic differentiation between those 2 localities. So we did something called nested clade analysis. We could examine how many times can we detect significant structure, either of louse populations between host species or between geographic locations. And for Phylogenomics, most of those were significant. Only 3 cases of not being significant. For Columbicola, all the cases we could look at, most of them were not significant. So if you just do an overall comparison of these ratios, Phylogenomics more often has significant structure either by host or by geographic location. So these body lice show more population structure as compared to Columbicola, reflecting the differences probably in their dispersal ability. So these differences in host specificity and dispersal ability might have implications for broad macroevolutionary patterns, such as the degree of congruence or similarity between parasite trees and host trees. So here's just a hypothetical example where we have perfect co-speciation. Every time host speciates, the parasite speciates as well. But what if we have a parasite that is not host specific? So if this parasite species E occurs on both D and E, that would break down that co-speciation event and the trees would be less similar to each other. So host specificity itself can influence the degree of similarity between host and parasite trees. But we need to reassess a little bit our definition of host specificity because we found these cryptic species, very divergent lineages within these different parasite species. So in some cases, Coevolution macroevolutionary is host specific. For this example, this macroevolutionary 3. In other cases, it's still widespread. And so when you average up, on average, Coevolution still tends to be on more host species than does body lice. So then we built phylogenetic trees for the doves that occur in pigeons and doves that occur in the New World, and also for their lice. And here's a comparison for the wing lice, which can disperse more and are less host specific. And although we could potentially reconstruct some co-speciation, it wasn't any more than we would expect by chance alone. Whereas for the body lice, we found a much stronger match between the trees. So there are many examples of co-speciation. One here between the white-winged and mourning dove and their lice. And this was much more co-speciation than we would expect by chance. So differences in dispersal ability correlate with differences in population structure, which influences host specificity, which in turn influences the degree of matching of host and parasite phylogenies. So pigeons and doves, as I mentioned, host both wing and body lice. Why do they have these two different groups of parasites? Well, it turns out wing lice have this long, thin body form, and they escape from host defense by inserting between the barbs. This is a wing feather, and these are the barbs. And they insert between this inner barb space to escape from host preening, similar to the feather mites. And that's probably why they have this long, thin body form. If you do a correlation, and this ecology of the way they escape from host defense also creates a macroevolutionary correlation between the width of lice and the body mass of the host. So bigger birds have bigger lice, smaller birds have smaller lice. So I've given you a couple examples here of macroevolutionary patterns explained by ecology, both in the match between host and parasite evolutionary trees, phylogenies, and the match over a comparative macroevolutionary scale of host and parasite body size. I want to turn to some even broader macroevolutionary patterns now in thinking about bird feather lice in general. So not only are wing and body lice in pigeons and doves, but other groups of birds, such as parrots, also have a body louse, this very rounded body form with a rounded head, these long and slender wing lice. But also some groups of birds, like parrots, have head lice that they escape from host preening by just remaining on the head, where the bird cannot reach its own head with the bill, so they're safe from preening in that case. And they have this rounded body form with a triangular head. And this pattern is repeated across many different groups of birds. So if we look at head lice, for example, on all different kinds of birds like ducks and hawks and so forth, we can see that they all look extremely similar to each other. Within a group of birds, there can be multiple representatives of these ecomorphs or niche specialists. So tinamous, for example, have head lice, wing lice, and body lice. So there's two possible scenarios for how these lice evolved. One is maybe they just evolved into these microhabitats early on during the course of bird evolution, and as birds diversified, the lice just diversified through co-speciation with them. In which case we would predict all the wing lice to be closely related and all the head lice to be closely related, and that to follow the pattern of bird diversification. Another scenario is these lice repeatedly diverge into these different niches within a group of birds. So maybe body lice started out on parrots, and then they diverged into head lice and wing lice. In that case, we would predict all the lice from parrots would be closely related with the head and wing and body lice. And then all the lice from tinamous, for example, closely related with head, wing, and body lice. So we tested this by building a phylogenetic tree based on just a few gene sequences in this case. So some of the tree was not very well supported, but there was a very clear pattern that emerged. So these are lice of different genera from different groups of birds. And what we found is several cases where lice from different microhabitats, but from the same group of birds, were closely related. So the wing lice here in red and head lice in blue from ducks were sister taxa. The head lice and body lice from parrots were sister taxa. And this pattern occurred way more than expected by chance, suggesting this is a real biological phenomenon of repeated divergence within a group of birds, but then convergence across the broader tree of lice, with all the head lice converging on the same morphology over and over again. So turning to even a higher scale of lice, in more generally, the traditional classification puts them in the order Theraptera, order of insects, with 4 major suborders. 2 of them, Amblyssa and Ischnocera, both occur on birds and mammals. So the feather lice I told you about from doves are in this group Ischnocera, but there's also some representatives from mammals. Then there's this suborder Rhynchotherino of only 3 species. It's a parasite of elephants and warthogs from Africa. These have chewing mouth parts, and so does this group. They have chewing mouth parts at the end of a long rostrum or snout that they chew through the thick hide of the elephant to get at blood. And this is thought to be the transition form to the true sucking lice Anopleura that occur on mammals, including humans. They have piercing sucking mouth parts, and they feed only on blood. And a morphological study by Chris Lyal in 1985 suggested that the Anopleura were derived from chewing lice. So some of you might know the term Melophaga. That was the order applied to chewing lice, but it turns out that group is paraphyletic, with sucking lice being embedded within it. And lice in general are derived from free-living insects in the order Psicoptera, bark lice and book lice. And so given the distribution of these major groups of lice on both birds and mammals, one question is, on what group of hosts did they first begin to radiate? Was it some ancestral mammal or bird, or maybe lice are old enough to have occurred on dinosaurs, which we know have feathers, so maybe that was an ancestral habitat. So to answer that question, we need to have some information about how old lice are, so we can get some of that information from the fossil record. Unfortunately, there's only one fossil of louse described a few years ago, but it's a really excellent specimen from the Eckfeld Mar in Germany, a compression fossil dating to about 44 million years ago. It's the first and only so far fossil lice, but it has really strong affinities with modern feather lice in the suborder Amblyssa. So this is the fossil, clearly in the family Menoponidae, and this is a modern slide-mounted specimen of the same family. And we can see the crop contents here. So the crop is where these lice store their food before it's digested, here and also here. And if you look carefully and squint a little bit, you can maybe imagine there are feather barbs in the crop. If you're paleontologists, love to do that sort of thing. But we only have one fossil calibration, but we can also take advantage of the fact that we know a lot about bird and mammal history and how old different bird and mammal groups are. So chimps and humans share a genus of louse that, when we know chimps and humans diverged about 4 to 6 million years ago, and so did their lice. And so we can use knowledge of this date to put a calibration point on this date. Okay, one of the problems for higher level phylogenetics is often we get trees that are not very well supported, particularly if we use more traditional methods of amplifying a few genes with PCR, so-called targeted sequencing, using Sanger technology. And usually when we find universal primers, lice we didn't know much about the genome, so it's very hard to design primers. So what we were stuck with is very non-optimal genes for phylogenetics. Either ribosomal genes that evolve slowly and need a lot of alignment, so there are issues there, or mitochondrial genes that evolve very fast and are not very appropriate for deep-level phylogenetic questions. And so it's difficult to design primers for a group for which you have no genetic information. And as you increase the number of genes, your PCR starts not working, and so you have a lot of missing data. And many of these deep phylogenetic problems we know are going to need lots of genes to solve them. And then one other problem is if we want to add species in the future, we have to sequence exactly the same genes that we started with, which might not be ideal. So I'm sure most of you are familiar with so-called next generation sequencing technologies. Illumina is an example of this. These are non-targeted approaches, so in theory you can sequence the entire genome, and they produce millions, but of fairly short sequencing reads. But the price per base pair is orders of magnitude cheaper than targeted sequencing, so you can get millions and millions of base pairs of sequence. There's a higher error rate than Sanger sequencing, but we rely on coverage, sequencing the same position over and over again to help us determine what the base should be. There are also some insect or louse specific problems that we have, and the same is true in mites. It's difficult to obtain a large quantity of fresh material that we might want to use in transcriptome sequencing, say sequencing all the expressed genes, which is a popular approach. Most of our material is also stored in ethanol, which completely degrades RNA, but also degrades DNA into smaller fragments. And from a single louse, we do not get much DNA, 5 to 20 nanograms, so it's not so optimal for hybrid capture or UCE type approaches. And because it's in ethanol, the DNA is degraded, so it's also difficult to make very long insert libraries that we might like to make for total de novo genome assembly. But lice do have some advantages. One is the genome is relatively small, 100 to 200 megabases, which is among the smallest genomes of all insects. And from a single specimen, we can get enough DNA for a single 300 to 500 base pair paired end Illumina library. So we can, in theory, sequence the genome at some amount of coverage from this type of library. And we have a reference genome now for the human head louse that we can use. So can we really sequence the full genome and use it in phylogenetics? And I would argue the answer is yes. We're doing this now for $400 to $500. We start out at about $1,000 per genome. So this price is coming down every day as technology improves. And we also need a method of assembling genes. And so we've developed this method called Target Restricted Assembly Method, or TRAM. It basically reverses the order of assembly and annotation. So first we compile a set of reference genes or targets that we're interested in assembling for phylogenetics. And then we use blast searches, tblastn, of an Illumina data set from about a quarter lane. This is where we're getting the price per genome. And then we assemble the matching reads locally. So we figure out what reads match our gene and then just assemble those together. And then we do some annotation to remove introns because they complicate things. And this process is now automated. My postdoc, Julie Allen, published this bioinformatics software, aTRAM now for automated, that we've been using. And so this is just an example of what this looks like when I was first starting out to see if this would work. Here are a bunch of Illumina reads from the chimp louse. We blasted the chimp louse library with a human louse sequence, got all these reads, and they assembled nicely together. We can get the consensus sequence by just taking the most common base. So this seemed to work. And so now we've applied this to phylogenomic questions in lice. So we've sequenced the genome of 47 louse species, including all major suborders and most of the important families, representing all the distinct bird and mammal host lineages to get at this question. For this project, we targeted a little over 1,000 genes for assembly using aTRAM and did sort of standard phylogenomics analysis. So this is the tree that we got. Nearly all the branches are supported by 100% bootstrap. There's a few exceptions. But one of the most interesting results that we got is Ischnosera, this widespread suborder, is actually paraphyletic. So there's a family here, Trichodectidae, that occurs on mammals. It's sister to the other groups that occur on mammals, Anapleura and Rhynchothyrina. whereas all the bird lice are up in this group. So we've done a molecular dating analysis of this tree as well, using these fossil calibrations and the human chimp split and a couple other calibration points. And what we find is there's these three main radiations of lice, one on both birds and mammals, one exclusively on mammals, and one, with one exception, almost exclusively on birds. And all three of these groups started to radiate as their modern hosts began to radiate at the Cretaceous-Paleogene boundary, the time of the mass extinction of the dinosaurs, and when birds and mammals really started to diversify. So this suggests their life began to diversify around the same time. Unfortunately, reconstructing the ancestral host was not as clear. So if we assume the ancestral host was a bird, we would come up with 4 switches to mammals, 2 within Amblycera, 1 at the base of this large mammal clade, and 1 to this weird louse. It's kind of like a bird louse on a mammal. It's on Madagascan lemurs. It's always thought to be an enigma. If we assume the ancestral host is a mammal, again, we get 4 switches to birds, 2 within Amblycera, 1 at the base of this major bird radiation, and 1 to this weird louse from a weird waterfall type bird called a screamer from South America. But all these radiations happen between about the Cretaceous-Paleogene and about 45 million years ago. So their switches are very old, and they've been very stable on birds and mammals since that time. So the conclusions here, we reduce the number of origins of mammal lice within lice by at least 1. Lice underwent simultaneous radiations of birds and mammals as the 2 groups diversified after dinosaurs went extinct. And there's 4 major host switches. We're a little bit uncertain in the direction, but they all occurred over 45 million years ago. Okay, just to highlight, in case some of you are interested in using this approach, some of the advantages of aTRAM. From these shotgun genome sequences, 1 library, the same data can be mined for additional genes in the future. So maybe we want to try a different set of genes and see how they perform. We could increase the number of genes or decrease the number of genes and assemble them again from the same starting raw data. So we're not stuck with the genes we started looking at. And adding taxa is straightforward and only needs to be done once. If you want to include a new species in your study that you hadn't looked at before, you just sequence the genome, and then you have all the data you would ever need. And these data sets can be mined for many applications beyond phylogenomics of the group you're interested in. So we're also assembling the genomes of microbial symbionts that occur inside these lice and finding all kinds of interesting results there. And we're also using these same data sets to assemble mitochondrial genomes. Lice have all kinds of strange gene rearrangements in their mitochondria. It's an interesting evolutionary question. Not the question we start with, but we can also gather that information from exactly the same raw sequence reads that we came up with. And with that, I'll just put up here a number of collaborators involved in all this work, and people that provided specimens and funding for this. It comes from the U.S. National Science Foundation and the University of Illinois. Thank you. I'll be happy to take questions.
Thank you. Fantastic talk. You have